Hey everyone, welcome back. In today's video, we're going to talk about POD5 files. Now, POD5 files are probably one of my favorite file types to work with because they have some interesting properties that can make working with this data a challenge. Let's get started. So what is a POD5 file? It's a file format for storing nanopore signal data, which are produced by Oxford Nanopore sequencing instruments. These files store information about DNA, RNA, and epigenetic information. And you'll know when you see a POD5 file because they end with a .pod5 file extension, which is an update to the legacy FAST5 file format. So where do POD5 files come from? Again, they're produced by Oxford Nanopore sequencing instruments. And the instrument on the left is called a Promethean. And what it does is it allows genetic material to pass through tiny holes in the sequencer called nanopores. And as the molecules are doing this, an electrical signal is emitted, which is captured by the device and written to a file called a POD5 file. So what is the POD5 format? You can think of a POD5 file like a database composed of several tables. The first is a reads table, which contains information about the molecules that pass through each nanopore. The second is a signal table, which contains the actual signal for each read that was measured from the device. And third is a run info table, which contains metadata associated with the experiment. And I find POD5 files kind of interesting because they have some special properties that require you to think carefully about how to manage and analyze this kind of data more efficiently. The first property is size. POD5 files are massive. In fact, the POD5 files produced from Promethean instruments can range between 700 and 2,000 gigabytes in size for just a single sample. So if you sequence just 24 samples, that's between 16 and 48 terabytes of data. That's insane. The second property is that base calling is still required. We have to remember, the POD5 files just hold the electrical signal data. We still need to convert these electrical signals to a nucleotide sequence that we can analyze. This process is called base calling, and we do this by using a software program called Dorado, which uses a machine learning algorithm to convert the electrical signals into a nucleotide sequence and writes this to a FASTQ file. Now, one of the interesting things about Dorado is that it requires access to GPUs, specifically high-end ones like the A100 and H100 NVIDIA GPUs. And this makes base calling a computationally intense and expensive process. This is a table showing how much it would cost to base call a single whole genome sequencing sample using different Amazon EC2 instances. For those of you not familiar with EC2 instances, they are essentially remote servers on the cloud with different hardware configurations. Different EC2 instances have different kinds of GPUs, different numbers of GPUs, and different prices. And base calling one sample can take between 1 to 53 hours and cost between 20 and 230 US dollars, depending on the hardware configuration used. And I think one super interesting thing about POD5 files is that you can hold on to them and base call them again once new base calling algorithms are released. And so far, I've only talked about base calling, but Dorado can also detect base modifications, and new ones get released as well. So what some people will do is archive the POD5 files in the cloud, and then after some time, redo the base calling and modification calling and perform the analysis again on the updated data. So where can you download POD5 files? That's actually a good question because they're pretty hard to find due to their size, the cost to store them, and difficulty sharing them. However, there is one repository called the Singapore Nanopore Expression Dataset that came out of the Genome Institute of Singapore earlier this year, which is hosted in the Registry of Open Data on AWS. The repository contains Oxford Nanopore datasets from 7 human cell lines that were prepared using different RNA library prep methods. And I actually use this data quite often for benchmarking my own base calling workflows on AWS, so make sure to have a look at this if you're interested. In the next video, we're going to talk about a very important file format called FASTA format, which is for storing reference sequences. You won't want to miss it. I'll see you there.